Hello, my dear students. Today the class is about a media formulation of that water and energy source. Myself, Dr. Shibu, from the Department of Biotechnology, Staff Institute of Advanced. So, in the previous class, what we discussed it is about media formulation, general aspects of media formulation, what is the role of media, and what is the major component of media which will support the fermentation industry. So, today, the class is about the major component in the media, water and energy. So, let's move to the topic. Water is a major component of almost all fermentation media and is needed in many of the ancillary services such as heating, cooling, then cleaning and rinse. Okay, so of course we know that in any media preparation, water is one of the major components. For example, water is the major quantity which we are adding in the preparation of media. Okay, not only in the preparing the media, and it also support as a material for cleaning, and for heating, then for cooling in the cooling jacket in the fermenter, and also for any other purposes. Clean water of consistent combustion is therefore required in large quantities from reliable permanent source. So, if you are running a fermentation industry, water is one of the predominant material which was support, and it should be a reliable and as also a permanent source. When assessing the suitability of water supply, it is important to consider pH and the dissolved salt, and also the effluent contamination. If you are using any type of water, that you need to consider about the pH and the dissolved salt. All the water is not same. You maybe tasted the water which is from your area, it is different, and in your college area, it is different. If you are moving from Malappuram to Ernakulam or to Trivandrum, the drinking water is different from the area based on their geographical location. The taste of the water will be changed because of the dissolved salt which is present in the water. Okay. So the mineral content of the water is very important in any brewing industry, and most critical in the matching process, and historically influence the setting of breweries and the types of beer produced. Okay. So the normally hard water which is containing high CaSO4, the high concentration of calcium sulfate, which is the concentration, if it is increased, it is suited for the production of English Burton bitter beer. For the production of English Burton bitter beer, we need to use high concentration of CaSO4. And where the water which is having high carbonate, which is suitable for the preparation of darker beers such as stouts. Okay. So nowadays the water must be treated by deionization or other techniques, and salts are added and the pH are adjusted to favor different beers, so that breweries are not so dependent on the local water source. So previously the water, we are not having any distilleries or deionization process, so that they will take water from the geographical location, so that they will prepare the breweries according to the water. Nowadays it is not like that, because they will, what they are doing is they are taking the water and deionized it. Okay, deionized, we will get distilled water. In that water we are adding the minerals, whatever you want you can add. If you want English bourbon beer, you can add high concentration of CaSO4. If you are a darker beer, you can add high carbonate content. Like that, they will adjust their ratio of the chemicals, and that water is used for breweries. Okay, because nowadays we cannot depend upon the pure water. All the ground bodies have been almost contaminated, so that they will distill it, and they are using it, deionize it, and they are using it. Okay, so that is about the water which is playing a major part in the fermentation industry. Okay, now the next is about energy source. Okay, it is energy sources which is giving an energy to the microbial cell for the growth, or supporting the growth. So energy for growth come from either the oxidation of the medium component or from the light. Okay, most industrial organism, most of the microorganisms are chemotrophs, which means an organism that obtain energy from the oxidation of reduced organic compound, that is called as chemoorganotrophs. Therefore, the commonest source of energy will be the carbon source, such as carbohydrate, lipids, and protein. Some of the microorganisms can also use hydrocarbons or methanol as carbon and energy source. Okay, so let's move to the carbon source and factors that is influencing the choice of carbon source. After before going to the carbon source, there are plenty of carbon source which is available as an energy source. So before moving to that, we need to know about the factors which are all influencing the choice of carbon source. It is now recognized that rate at which the carbon source is metabolized can often influence the formation of biomass production of primary or secondary metabolites. Okay, so what is mentioned here is that the rate of the carbon source which is metabolized, if it is metabolized quickly or slowly, depend on that, it will influence the formation of the biomass as well as the primary and secondary metabolic formation. Fast growth due to high concentration of the rapidly metabolized sugar is often associated with the low productivity of secondary metabolites. So it is noted that if the sugar is rapidly metabolized or fast metabolized, what will happen, the growth of the organism is also very high, and suddenly it will attain the stationary and the death phase, and it leads to the depletion of the secondary metabolite. So the fast growth of the organism is very high, and the sugars are rapidly metabolized, so there is no carbon source is there, so there is a decline phase, so automatically there is a low productivity of secondary metabolites. In such cases, that problem may be overcome by using a less readily metabolized sugar such as lactose. Instead of directly adding a glucose to the media, you can add the lactose, which will take time for metabolism. Okay, so that's what it is mentioned. Next point is the main product of the fermentation process will often determine the choice of carbon source. Okay, particularly if the product result from the direct dissimulation of it. So okay, so the main product of the fermentation process which is depend on the choice of carbon source what we are going to prepare. That's what it is mentioned. In fermentation such as ethanol or single cell protein production, where raw material are 60 to 77 percentage of the production cost. The selling price of the product will be determined largely by the cost of the carbon source that we are using. Okay, so in the fermentation of ethanol and single cell protein, it is mentioned that the cost of the raw material is 60 to 77 percentage, so that you need to sell the product in a higher price. That's what it's mentioned. It is often part of the company development program to test a range of alternative carbon source to determine the yield of product, and it influence on the process and cost of producing biomass and other metabolic. Okay, so there is a program which is having in all the pharmaceutical companies which is having an alternate carbon source. There is a research wing is there. They are checking for an alternate carbon source which is very cheaper to produce a biomass in a large scale. If it is produced, we are using an alternate carbon source which is commonly very cheaper than rather than they are using a highly costly carbon source. They can able to use a very cheap carbon source so that they can sell the product in a very cheaper amount, so that they'll get a benefit. So the pharmaceutical companies will get a major benefit. That's what. This enables a company to use alternate substrate depending on price and availability in different location and remain competitive. So this carbon source, the utilization of the carbon source, the alternative substrate just depend on the price and the availability in a different location. So based on the location, geographical location of the pharmaceutical and other fermentation industry, they need to check the available carbon. So okay, the purity of the carbon source may also affect the choice of substrate. For example, metallic ion must be removed from carbon sources used in some citric acid production. So the purity of carbon is also another question mark. You need to use a proper purified carbon source, otherwise it will disturb the downstream process. Okay. So for example, metallic ion must be removed from carbohydrate source which is used in a, some citric acid process. Then the method of media preparation, particularly sterilization, may affect the suitability of some carbohydrate for individual fermentation process. Sterilization is also another thing we need to take into account because sometimes the sugar should be sterilized separately, and other metals should be separated. Sorry, it may be sterilized separately. because it will lead sometimes it leads to a caramelization. Sugar which is present in this carbon source leads to caramelization, so that sometimes they will sterilize this sugar separately and other compounds separately, like that. The media sterilization, and this is also a factor that was influencing the carbon source. Okay, so hope you will understand with this water and energy source, particularly about the factors which is influencing the carbon source, and the uh, remaining things we'll see in the next class. If you have any queries, you can contact me. So that's all for today. Thank you.